All right, welcome back to part 2 of transcription. By the end of this lesson, I hope that you will be able to describe the characteristics of a bacterial promoter, explain how RNA polymerase transcribes a bacterial gene, and compare and contrast the two mechanisms for transcriptional termination in bacteria. So, our molecular understanding of transcription all originated with E. coli. Again, this is where we tend to start, but we've expanded from there greatly, and most bacteria utilize a similar type of system. Now, promoters are simply DNA sequences that promote gene expression. And more precisely, they direct the exact location where the initiation of transcription will happen. So if you recall, transcription has three phases. It has initiation, elongation, and then termination. And this is essentially setting up the site where the initiation will begin. So promoters are a DNA sequence that exists in the genome. It exists in several locations in front of each gene. And we'll get this process started. Now, these promoters are typically located just upstream of the site where transcription will actually begin. So, if we take a particular gene and we have the start location of its transcription, we'll call that +1. And everything downstream of that will be called +2, +3, +4, +5. However, far it goes. When we refer to the region upstream or preceding it towards the 5' end, in front of whatever gene was there, we will start counting backwards, but we will skip 0. So we go directly from +1 to -1, and then -2, -3, -4, -5, and so on and so forth. There are two specific regions, the -35 sequence and the -10 sequence, which directs this process in bacteria. And together, these two sequences are called the promoter region. And another name for this would be the core promoter. Now, the sequences that exist here can vary slightly. It can vary slightly between organisms and even within an organism. And we'll talk about that in a moment here. Also, the exact locations can vary by a nucleotide one way or the other. So in one particular gene, this might not be the actual -35 sequence. It could be the -36 sequence or the -34 sequence. But you get the idea. We've got these general sequences that are going to occur in these approximate spots. So these sequence elements are actually serving as a binding site for a protein that will come in and recognize these sequences. So the protein that recognizes it, the closer it is to the perfect sequence, the more interactions will occur and the tighter it will bind. Now, in addition to referring to this as the -35 and the -10 sequence, this first -10 sequence is also sometimes called the Pribnow box after its discoverer, but you can really call it whichever. But this provides a binding site for a protein that will allow transcription to start beginning at this +1 site. So here's the +1 is an A on the coding strand. On the template strand, it would be a T. So when we make a messenger RNA, it will be an A. Here is an example of many different sequences from all within E. coli, different gene loci. And you can see here that the sequences are not exact. Some of them are very divergent. You can also see that the spacing between them are somewhat variable. So here we have 17 nucleotides in between. Here we have 16 nucleotides in between. Over here we can see 6 or 7. If you take all of these sequences together and look for the most common nucleotide in each position, you get what we call a consensus sequence. And this consensus sequence represents the sequence that the protein that binds this will bind the tightest to, meaning that it has the most interactions along the spacing that will allow it to associate very tightly. All right, so we now understand what a promoter is. Now let's take a look at RNA polymerase and how they work together to initiate transcription. So RNA polymerase is the enzyme that catalyzes the synthesis of RNA. And just like DNA polymerase, it is a holoenzyme that is composed of several subunits. We can break these subunits into two key parts. So we have the core enzyme, which has 5 subunits, 2 alpha subunits, a beta subunit, a beta prime subunit, and the omega subunit. This is the core part that is involved in actually making RNA, and this is the part that will continue on making the rest of the RNA chain as it progresses on through elongation. The sigma factor is made up of a single subunit, and this is actually what binds those -35 and -10 sequences. And each subunit has its own distinct function and role. Now, you'll notice here that we have brought up, yet again, more Greek characters. So just for kicks and giggles, we'll go ahead and throw in a chart of the Greek alphabet. You might want to just briefly familiarize yourself with it. You certainly don't need to memorize it, but you'll see these come up again and again and again. To give it a historical context, when a lot of these things were first discovered, knowing the Greek alphabet would have been normal for a science student. It's very heavily used in mathematics and in physics and many other areas. So utilizing the Greek alphabet is fairly typical in all of sciences. All right, so the RNA polymerase holoenzyme is going to loosely bind to the DNA. And this has the ability to move forward and backwards along that DNA, kind of like a shower curtain on a shower pole, right? So it can just move back and forth, or curtains on a curtain rod. It will move down the DNA until it encounters a promoter region. When it does encounter a promoter region, the sigma factor subunit will bind the -35 and -10 regions, and it will cause the holoenzyme to pause. That pausing activity is what allows the holoenzyme to start the initiation process. So this sigma factor is a protein that has a helix-turn helix structure. This is what that looks like. So there's an alpha helix followed by a short turn and another alpha helix. And you can see how these helices fit nicely into the major groove. It just lays right in there. And if you recall, in an alpha helix, all of the amino acids point outwards, or the side chains point outwards. allowing them to make contacts with the individual base pairs. So here we can make several different contacts with several different nucleotides. The overall process of how this works, as I said, it is clamped around the DNA in a way very similar to the DNA polymerase. It'll just be moving along until the sigma factor encounters the promoter region. It will bind to the promoter region and cause it to pause. At this point, the DNA is closed or still wound up in an in a helix. So we call this the closed complex. But this pausing activity allows the machinery of the RNA polymerase to act on the DNA, opening it up, creating an open complex. Once you have an opening, then RNA nucleotides can start coming in, base pairing, and the catalytic mechanism can kick in, and you will start transcribing. At this point, the sigma factor falls off, and it will allow the holoenzyme, or the rest of the core polymerase, to travel on without that sigma factor. And this would mark the end of the initiation phase. Past this point, the RNA polymerase would continue to make RNA transcript, and this would enter into what's known as the elongation phase. Now, the RNA polymerase, as it moves along and is synthesizing the RNA, this is the stage that we call the elongation stage. And be sure to keep the DNA strands in mind that we are using. So what we're making is a copy of a coding strand. So we will use the antisense strand, or the template strand, in order to add the nucleotides onto. The opposite strand, the coding strand, will be pulled out of the way, and the sequence will be the same on the coding strand as it is in the RNA transcript, except for that, of course, whenever you have a T in DNA, that will be added in as a U in RNA. So here's kind of a close-up picture of this open complex of the core region moving along a transcript during elongation phase. You can see the 5' end is hanging out. We are adding on to the 3' end. So net, we are moving from a 5' to 3' direction. Normal synthesis. There is a bubble that is opened up inside, what we refer to it when we say the open complex, we're referring to this bubble. That bubble is about 17 base pairs long, and as we add in new nucleotides and move forward, the end of the RNA will stick out the back, and the DNA will collapse back in on itself and refold into a helix. This is something that can move very quickly. It can add about 43 nucleotides per second, so it can really zip along. Both strands of the genomic DNA are used as a template strand for different genes. So here we have an example of 3 genes, gene A, gene B, gene C. And you can see here that both gene A and gene B are running off of one strand. So here we have a promoter, gene A, terminator. Promoter, gene B, terminator. And the top strand of the DNA shown here would be an example of the coding strand and coding strand. And the template strand is going to be this bottom strand. And what you will end up with is an RNA that is a copy of this upper strand. So transcription is going this direction. Gene C, however, is in the opposite orientation. So it has its promoter, then the gene, and terminator here. What that means is for gene C, the bottom strand is the coding strand, the top strand is the template strand, and when you get an RNA from it, it'll be a copy of this lower strand here. So we have genes in all different orientations throughout our genomes, and bacteria have different orientations throughout their genomes as well. On occasion, you can even have genes overlapping. So a promoter for another gene might actually be part of the coding region for another protein. So you can have a lot of variety of how this is set up. Um, Probably by far the most complicated version of this is what you see in viruses. Viruses are simply a piece of genetic material encased in a protein sheath. And the smaller their genome is, the easier it is for them to transmit it. So consequently, viruses tend to have multiple genes overlapping in both directions. Now, termination is when you end the RNA synthesis. Without some type of process in place, that RNA polymerase would just keep on going on and make an infinitely long RNA. So there's a process by which we can knock it off. And E. coli has two different mechanisms for this. The first one is what we call a row-dependent termination, and it requires a protein known as Rho. The other one is called row-independent termination because it does not require Rho. So let's take a look at how this works. So first we'll go through row-dependent termination. So in the sequence of DNA, we have a site that is the row recognition site. That is transcribed in the growing messenger RNA. So here's the template up here, it's transcribed, and now you have this rut site present on the messenger RNA. That rut site recruits the Rho protein. So Rho will assemble, and Rho is a helicase, just like the helicases we see on DNA during replication that bind around single-stranded DNA. This one is just specific for RNA and assembles around this rut site. So as soon as it assembles, it will start motoring up the messenger RNA, and provided that it never catches it, nothing will happen. Well, also in the sequence of DNA, there is the coding for the formation of a stem loop in the messenger. So within the DNA, there is a complementary sequence here and just downstream here that will cause the stem loop to form. This stem loop will bind the polymerase and cause the whole thing to pause. That pause allows the Rho protein to catch up and then disassemble the complex. So you can kind of think of it as this is a loading site for like a little messenger RNA missile that assembles on and then shoots up, and it normally could not catch the polymerase, but the stem loop causes it to pause, and that will knock the whole assembly off. Now, Rho independent has a slightly different mechanism. So here, there are two sequences that are of importance. One is a uracil-rich region located at the 3' end of the RNA, and this also has a stem loop structure. This time it is upstream of this particular sequence. So in Rho independent termination, here we have a growing messenger RNA, it will form a stem loop. And again, this stem loop will cause the polymerase to pause, just like we see over here. So we get a pause, and then this U-rich region is slightly weaker in association. So the hydrogen bonds that you form between uracil and adenine are slightly weaker than what you encounter normally, and it has to do with the bond angles. So the fact that you have a U-rich region means that this sequence is slightly destabilized, and it will tend to fall off when you reach this pause. So now we have row-dependent termination and row-independent termination, because here we do not need row. Here we need row, and row is physically knocking the polymerase off.